Hello students, how are you people? I've come with a new topic for you people today, and we are going to discuss enzyme inhibition. Inhibition of enzymes. This is a topic of class first year, and I have already uploaded lectures about enzymes, introduction of enzymes, the properties of enzymes and all these things. You can go to my previous lectures and you can watch the videos, you can attend the lectures again. All of that is present for you people. I feel very happy when my students come to my channel and they watch the lectures. Today we are going to study about enzyme inhibition. This is a very interesting topic and I really like this topic. Enzyme inhibition. Inhibition means something to stop. Inhibition, when we talk about inhibition, when we talk about inhibitors, when we talk about inhibit, it means that something, stoppage, that is causing something to stop. That is known as inhibition. Enzyme inhibition is a factor that is, you can say, kind of stopping the normal enzyme activity. That is stopping the normal enzyme activity. That is known as enzyme inhibition. And the chemicals that bring about this inhibition or this stoppage into the enzyme activity are known as the inhibitors. They are known as the inhibitors. We have inhibition which can be permanent. We have inhibition which can be temporary, which can be reversible. We have inhibition which is permanent. Permanent inhibition is that type of inhibition which brings about a permanent change, and such type of inhibition is said to be irreversible. It is said to be irreversible. You can get a question in the exam that you have to write a note on inhibition, and there is another type of inhibition which is reversible. The effect can be reversed. It can be returned back to the normal. Inhibitors are actually special types of chemicals which kind of they resemble the original substrate of the enzyme, and they kind of cheat the enzyme. And the enzyme thinks that this is the substrate, and then it attaches the substrate to the binding site, and then in that case what happens, it blocks the binding site. Now this is a case where the substrate and the inhibitor are competing for the active site. They are competing for the what? For the active site. Active site is that area where the reaction is going to take place, where they are competing for the active site. Such an inhibition is said to be competitive inhibition because they are competing for the active site. The substrate kind of resembles the inhibitor resembles the substrate, and then it attaches to the active site and it blocks the active site. This can be reversed by increasing the concentration of the substrate, and then the inhibitor will not get the chance to come close to the active site. But however, this type of inhibition is said to be competitive inhibition, and the inhibitor that is of this type is said to be the competitive inhibitor. It is reversible, and a very good example of this type of inhibition is the malonate. Inhibitor of malonate. Malonate, what does it do? Succinate dehydrogenase enzyme that catalyzes the formation of fumarate from succinate is competitively inhibited by malonate. We will see the sketches in that. The importance of competitive inhibitor is that it supports the lock and key hypothesis, and it shows that substances which are similar to the substrate are not acted upon by enzymes. Even if they get attached to the enzyme will not react with them. The enzyme will just be compatible with them for the binding site, but the catalytic site will not work on the competitive inhibitors. The competitive site is not going to change. It is not going to change the inhibitor into anything close to the product. The inhibitor will just stay as it is. This is one thing that you have to, that is, it is of importance and you have to keep in your mind. Competitive inhibitors are used as drugs in the control of bacterial pathogens. This is a good use of competitive inhibitors. They are used in the form of drugs for bacterial pathogens and antibiotics and etc., because they combat the bacterial infections, and they cheat the bacterial enzymes, and they get attached with the bacterial enzymes. Keep one thing in mind, then competitive inhibition is a little bit, it is a kind of temporary inhibition which can be changed by increasing the concentration of the substrate. Now, the next type of inhibition that we have is that inhibition which is not competing for the active site. In this case, this is known as the non-competitive inhibition. The non-competitive inhibition. This non-competitive inhibition is actually that type of inhibition in which the active site is not going to be involved. The substrate does not care for the active site. It attaches to some other site. The active site is not involved in this case. It is not involved. The enzyme attaches at some other site like the allosteric site. It gets attached to the allosteric site. It brings about a change in the shape of the active site. And when it brings about a change in the shape of the active site is available, but it cannot attach the substrate because the shape has changed. This is a kind of a permanent inhibition. It is a kind of a permanent inhibition. In some cases, we have the reversible non-competitive inhibitors, but in most of the cases, we have a permanent non-competitive inhibition in which it is irreversible. And the enzymes, they are permanently de-shaped, they are permanently denatured by the attachment of the inhibitor. In this case, what do we see? In the non-competitive inhibition, what happens that the inhibitory molecules, they do not attach on the active site. They attach on the allosteric site. These non-competitive, they can be reversible, they can be irreversible. The reversible ones are actually those ones in which they just attach on top of the enzyme and then they prevent the formation of the enzyme product complexes, and they prevent the conversion of the substrate into the product. The substrate, it gets attached to the enzyme, but it cannot be converted into the product. But this, however, is a reversible situation. On the other hand, the dangerous situation is the irreversible situation, in which a non-competitive inhibitor, it alters the site of the shape of the active site, so that the substrate is unable to attach to the active site. And a good example of this one is the cyanides. Cyanides are poisons. They are poisons of living organisms because they can kill by inhibiting the cytochromes of the cellular respiration. Cytochromes are special types of enzymes, cytochrome oxidases, which are present in the electron transport chain of the respiratory system. They block these enzymes and combine with them, and then they destroy the prostatic groups that are present inside them. And then what happens? They break down the enzymes, they break down the disulfide bridges, and they totally destroy the tertiary structure of the enzyme, and they disrupt the structure of the enzyme. That is in the case of the permanent inhibition, permanent non-competitive inhibition. Let us go to the presentation and let us see what we have in the presentation. First of all, the definition of an inhibitor. What is an inhibitor? Inhibitor is a chemical substance which can react in place of a substrate with the enzyme, but it cannot be transformed into the product. This is the very important point. It blocks the active site for some time, or it might be permanent. Permanent in the case when there are poisons like cyanides. And some of them, these cyanides, they destroy the shape of the active site. These inhibitors can be permanent, they can be temporary. Antibiotics, antimetabolites, these are also examples of inhibitors. We have two types of inhibitors, those that are irreversible in nature and those that are reversible. The reversible inhibitors, the main character of the reversible inhibitors is that they form weak linkages with the enzyme. They do not form very strong bonds with the enzyme. Their effect can be neutralized completely by what? By just increasing the concentration of the substrate. I have told you this already. By increasing the substrate concentration, their effect can be neutralized. What are we talking about? We are talking about reversible inhibitors. Then we come to the irreversible inhibitors. They check the rate of the reaction by either permanently occupying the active site or by destroying the globular structure of the protein, globular structure of the enzyme. They occupy the active site not by weak bond, by covalent bonds, and they may physically block the active site in the case of irreversible inhibition, or they might attach permanently to the allosteric site and destroy the shape of the enzyme. The reversible inhibitors, they can be divided into two major types, the competitive and the non-competitive inhibitors. The competitive inhibitors, because of the structural similarity with the substrate, they may be selected by binding sites, but they are not going to be changed into the products by the catalytic sites. Malonic acid competes with succinate for the active site of succinic dehydrogenase. Now, this is the situation. Competitive inhibition, this is the active site. This is a substrate molecule that is occupying the active site. This is the enzyme molecule. Now, this is the inhibitor, which is kind of resembling the substrate. It gets attached to the binding site, but the catalytic site does not accept it. Non-competitive inhibitors, they form enzyme inhibitor complex at a point other than the active site. They alter the structure of the enzyme in such a way that even the genuine substrate, it cannot bind to the active site. Why? Because the shape has changed. Now, this is the situation. This is the enzyme molecule. This is the substrate. This is the allosteric site. The inhibitor comes and attaches to the allosteric site. It deshapes the enzyme active site. The genuine substrate is attached, but the reaction will not take place because the three-dimensional shape of the enzyme has changed. Now, this was all for the lecture today. What we will do in the next lecture is we will be studying about the classification of enzymes. And see you people next time with the next lecture, classification of enzymes. I hope it is clear to you people. See you people next time. Take care of yourself. Goodbye.